Hello and welcome to this AIM webinar on prospects for a vaccine for COVID-19. AIM is an alliance enabling the cooperation, development, and standardization of AIDC technologies. From barcodes to RFID to IoT, AIM's your advocate. Any organization with an interest in data collection is a beneficiary of these efforts. Membership provides an opportunity to influence the direction of the industry, develop policies and standards, access market reports, and engage new partners. Before we get started, there are a few housekeeping items I would like to go over. First is our antitrust policy. It's the policy of AIM Inc. to conduct its operations in strict compliance with the antitrust laws. No AIM activity shall create even the appearance of a violation of the letter or spirit of the antitrust laws. Next, we have AIM's collaboration and work product policy. This states that presentations like today's are held for the primary purpose of advancements in our industry, which necessarily involves development of work product intended solely for the public domain. AIM's developed this policy for the protection of its members who engage in this important collaborative effort. I also want to note that attendees are muted throughout the presentation. If you have a question, our presenter will be answering some of those at the end of the presentation. You can ask questions by chatting those to AIM member services. A recording of this presentation will be made available and attendees will be notified when this recording is available. Without further ado, I would like to welcome longtime AIM contributor and expert in all things AIDC, Clive Hoberger. Clive, take it away.
Okay, great. Well, thank you. For those of you who have known me for many, many years, you lived through my life. I saw Jack Kinsbotter there. I was formerly a Zebra vice president until I retired in 2008. And at Zebra, I developed the ZPL programming language. I designed and developed it. I developed the first Zebra thermal transfer printers and started the Zebra supplies and the Zebra RFID businesses. Most of my patents are actually in RFID. And as my last act, I developed the ultracode color barcode symbology, which was issued by AIM in 2016. I've been a member of AIM since the Upper Paleolithic period in 1982, former chair, and I received both the AIM Dilling Award last millennium and an AIM Career Life Achievement Award. Now, why am I talking about medicine? The other half of my career was actually in medicine. Prior to joining the AIDC industry in 1982 as VP of R&D of Weber Marking Systems, I'd spent 12 years in biomedical engineering at Brookhaven National Lab and Abbott Laboratories. In my postdoctoral work at McGill University, Montreal Neurological Institute, I developed the first computer-assisted brain vascular surgery technique using a PDP-12 for those of you who may remember it. And while I was at Zebra, I became editor of the ISBT 128 standard for blood products. And Bruce Ray of Computype and I were honored by WHO in 2019 at the Lisbon meeting, when this standard was extended for all medical products of human origin, everything from cells, tissue, organs, breast milk, etc. And in 2012, I was given the One World Award for a career life achievement in blood banking IT. So after I retired from Zebra in 2008, I went back to medical technology, developed the first RFID operated blood bank at Blood Center of Wisconsin. This was FDA approved. And I've since then been primarily collaborating with Mohammed Abdu at Temptime in development of temperature sensing barcodes for the WHO vaccine labeling. And WHO set standards in vaccine, and they will be important in the COVID-19 vaccine labeling. So I'm about to commit an act of journalism. This is not a medical discussion. It is a discussion about a very remarkable process that's being used to develop the COVID-19 vaccine. All the information used here is publicly available over the internet. And I am not a physician or a life scientist, and I'm not qualified to give medical or scientific opinions about the vaccines themselves. And neither am I endorsing any technology, vaccine, therapy, or company. And as an all medical presentation, I need to disclose the fact that I do own less than $50,000 stock in 4 of the key companies involved in COVID-19. 2, Pfizer and Moderna, and directly involved in the vaccine development. Now let's talk about coronaviruses and COVID-19. COVID-19 is a mutation of a family of coronaviruses that are all related and cause diseases in mammals and birds. And it is closely related to 2 more deadly diseases. SARS, severe respiratory syndrome, and MERS, Middle Eastern respiratory syndrome, which you probably haven't heard of. There is evidence that it was a mutation that incubated in bats and pangolins. And if you've never seen a pangolin, it's a scaly anteater and it has scales that make it look like a giant avocado. Oh, excuse me. Yeah, not avocado. Artichoke. There was a opinion rendered by a committee of WHO this week that the primary mode of transmission is human to human, and it's in aerosols, which are spread by breathing, sneezing, singing, or coughing. Let's talk a little bit about epidemiology, which is the science of epidemics. The COVID-19 pandemic today is very similar to the Spanish flu pandemic of 1918-19 in a number of ways. Spanish flu, on the other hand, was an H1N1 influenza virus. We think, because the records are very difficult to find, that the death rate was around 10%. We know what the virus was because it was obtained from corpses in an Eskimo village where all the bodies have been frozen since 1918. Both pandemics have a similar characteristic in that there was no cure or vaccine available at the start of the pandemic. And also, President Wilson basically tried to suppress all the information about it because we were still in the middle of World War I. COVID-19 death rates are locally between 4 and 16%, much higher in Europe during the European portion of the pandemic. But the cumulative overall death rate so far is about 4.7% worldwide. And the US is running just about that, a little less than that. The principal ways you handle this type of epidemic, where you have no cure or vaccine, are socially distance people to prevent spread, prevent infectious contact. And just one person wearing a mask in a 2-person group reduces the spread by potentially 70%. 2 people about something like 8% to about 8%. The other thing you do, and this has been done since biblical times, is test, trace, and isolate the infected people to prevent further spread. And as we've learned over the last 6 months, we've had a lot of experimentation and in trying to find effective treatment to cure the infected. We no longer just automatically put people on respirators, but we also recognize that there are about 4 modes of death and try and treat all of them for the patient. It's not just necessarily a lung infection. The only way to keep long-lasting immunity is to create what's called herd immunity, where when you have enough of a population that is vaccinated, then the infection won't spread through that population. Typically, it's around 80% for something, for most viruses. And creating herd immunity basically requires vaccination here of probably around 80% of the population. How do vaccines work? Basically, the purpose of a vaccine is to generate an acquired immunity in a person to a particular disease. And it's very interesting how old vaccination is as a concept. The first vaccine for smallpox was developed by Dr. Edward Jenner in 1798 in London. And he observed the fact that people that got cowpox, which was a very mild form of a virus, didn't get smallpox. And so he administered live cowpox virus to people to stimulate them to generate antibodies to the deadlier smallpox. And that was the beginning of the concept of vaccines. Now they use inactivated viruses, but it has the same result. Vaccines basically contain a non-infective proxy agent that uniquely resembles at the cellular level, the disease-causing pathogen. The proxy agent then stimulates the immune system to create antibodies. And the purpose of having those antibodies are to destroy any future pathogens which might be encountered or with which you might be infected. The developing a vaccine in the past has traditionally been a multi-step, multi-year process. One of the first things you want to do is find a mutation-independent feature of the pathogen, be it a virus or a bacteria or a mold spore or anything else, and create an antibody to attack it. Developing a proxy then to stimulate the immune system to make the antibody, and then taking that into 3-phase clinical trials, and then approval in the US by the FDA and in foreign regulatory environments as well. And then typically people start manufacturing and distribution. For COVID-19, because of the seriousness and worldwide nature of the effort, the objective is to have an approvable vaccine on the shelf by the end of 2020. It's a very lofty objective. And to do that, all processes above must be accelerated and overlapped. Now, this has been a very unique process. WHO is an arm of the UN, which is basically an organization set up for communication, collaboration, and coordination of medical information. And it is designed to deal with issues like pandemics. And it acts as a clearinghouse for information. We were extremely fortunate that the Chinese scientists in Wuhan released the full DNA sequence of COVID-19 virus on January 10th. At that point, the first vaccines were developed, and the first vaccine went into phase 1 clinical trials 66 days later, not 2 or 3 years, 66 days. Also, the FDA developed a special program called Fast Track to cut the approval time to less than 60 days. This is different from the FDA's compassionate use program. This is actual regulatory approval. And that is now to be used on all the vaccine candidates. Okay, there are three phases to clinical trials. Okay, this was a system for testing that was developed by the British Medical Council in 1930 and is now used worldwide. And because most of the companies are global companies and many of the organizations deal globally, everybody basically uses the same clinical trial method. The phase one is what's called safety screening. Safety, the issue is, if I inject this into a patient, will there be side effects? Will there be totally unknown side effects? Is it safe? Will any patients die? That's usually fairly quick, and nowadays, since we pretty well know what the human immune response is going to be, it is perfunctory and typically doesn't reveal any major issues. The phase two is now where you're doing two parts. One, you're doing a medical proof of concept. You take a group of patients, you find out, do they actually produce antibodies? Okay, that's phase one. It's your proof of concept. Phase two, basically you look at what the immune response is as a function of the dose, so you can find out at what dosage you get good response and at what dosage you start to get adverse effects, side effects. Phase three is the most important phase, and in the case of vaccines, typically is about 10,000 patients or more. People generally aim for 30,000, but that's not always achievable here. Here you take already infected patients, okay, and you do a double-blind study against placebo so that when the results are analyzed, you can tell if the vaccine had any differential effect against other methods of therapy and against the placebo. This is the confirmatory step necessary for regulatory approval. Let's look at it. Let me back up. This whole effort is very unique in terms of the fact we have traditional and very non-traditional technologies for developing vaccines under tests simultaneously. Okay, there has been so much progress in genetics that we are now looking beyond the traditional inactivated virus to stimulate the immune system into things where we're using viral DNA and recombinant DNA. And the most modern technique, okay, which was pioneered by really three groups, Moderna Pharmaceuticals in Cambridge, Mass, the National Institute of Allergy and Infectious Diseases, which is headed by Dr. Fauci, and a very brilliant group of scientists at Imperial College London. The whole idea is to use messenger RNA rather than DNA. If you think of DNA as the cell's hard drive, messenger RNA is the USB drive that communicates the program to create the the spike protein. And two things happen. Once you inject messenger RNA into a body, its own cells produce the proxy antigen for the spike proteins. And if you do everything right, it also produces more messenger RNA. So you get amplification of the messenger RNA as a byproduct of producing the spike proteins, which the immune system then creates antibodies for. It's a revolutionary vaccine testing. There are 5 collaborations going on. I'll discuss which later, but they're going on primarily in the US and Europe and China. And there will be huge advantages in manufacturing if this technology works, and I'll discuss those in a later slide. Now, WHO publishes about every 10 days a landscape of the vaccines in clinical and preclinical trials. And the URL is at the bottom for those that are interested. There are 149 candidates, of which 17, I believe, or about 17 are in clinical trials. The most advanced one is the non-replicating viral vector, which is again a variation of the traditional technique for developing vaccines. It's developed at Oxford University and in collaboration with AstraZeneca, the big Swedish-British firm, and it's being supported by the US Biological Advanced Research and Development Agency as well. The trials, phase 1 and 2, are very good. The UK trials has run into a problem. The problem is that the infection rate in Britain has dropped so bad that they're now having to expand into hot areas like South Africa and Brazil in order to find enough sick patients. This is going to be a testing problem that we are going to see throughout all these various testings. And in the left, I just identified, highlighted one word that code word we can use for each one. The CanSino Biologics program with the Beijing Institute of Biotechnology is extremely important. We had phase 1 and 2 trials in China and Canada, but last week, the People's Liberation Army Medical Research Division gave a 1-year approval for use by the Chinese military. This is not the same thing as the Chinese government medical authority giving approval, but they have so much confidence in it that it's going to be administered to the Chinese military immediately, even though it's still going into phase 3 trials. Moderna is working, I pointed out, with the division of the NIH in this radical new technology using messenger RNA. Phase 2 is pretty well completed. They did studies in both patients under 55 and over 55. Phase 3 has just started. They're hoping to get up to 30,000 patients. Where they get 30,000 patients may be a bit difficult, but remains to be seen. They'll probably end up doing some testing overseas. Now, the Wuhan Institute of Biological Products, who did, I believe, the original DNA sequence, is working with a very large Chinese company called Sinopharm. So is the Beijing Institute of Biological Products. The Wuhan one is more advanced. 100% of the patients developed antibodies with 2 injections, and they're about to start phase 3 in the United Arab Emirates, where they can find probably at least 10 to 20,000 patients. Sinovac is another large Chinese company that specializes in producing vaccines, and they are in phase 2. It's again an inactivated virus. Phase 3 is going to be done in Brazil, which is basically infection central. And it will be done at 12 research centers in 6 states of Brazil. And it should be, they hope to complete it by the end of the year. Novavax, who you may have seen in the news yesterday, is a company Another US company in Baltimore, a spin-off of Johns Hopkins, and they're working with a company called Emergent Biosolutions, which is a company that develops custom drugs for the Department of Defense and others, primarily against potential chemical warfare agents. And so they're very good at developing things, but they do not have large manufacturing capacity. Novavax has bought a Czech company called Praha Pharmaceuticals, and they are looking for partners, and they'll probably team up with a company like Merck. They're testing both in Australia and in the US. The German effort also focused on messenger RNA, and they have allied themselves with two huge pharma companies, Fosun Pharma, which also owns Sinopharm, and Pfizer in the US. This is a messenger RNA. So far, things went very well in phase 1 and phase 2. Pfizer has taken over the management of phase 3, which will start by the end of this summer. Additional inactivated viruses are being developed at the Chinese Academy of Medical Sciences, but they have a unique approach which they've used in development of a polio vaccine, and we don't know a lot about what the trials are. The last company is Inovio Pharmaceuticals, which is working with the South Korean NIH, and it has a very unique approach where they use what's called a plasmid, which is a section of DNA, and it's injected, and then they use electrical stimulation to get it to be reproduced in the cells and to create antibodies. Very unique approach. There are 7 additional candidates in phase 1, including 2 other messenger RNA, and there are 132 candidates additionally in the preclinical phase. Now, someone might ask, why are so many people developing vaccines? Well, I would use the analogy of the great bank robber, Willie Sutton, who was asked why he robbed banks. And his answer was very simple, because that's where they keep the money. Obviously, this is going to be hugely financial. It's going to add probably at least $20 to $30 billion to the worldwide vaccine market. Now, a key question that we all probably would ask ourselves, wait a minute, we're hearing this vaccine is mutating. And if the virus mutates, will the vaccine still work? Or will it be like flu vaccines, where this year's vaccine may not work with next year's mutation? Well, COVID-19 originally mutated from a virus probably in bats, and it mutated actually in Wuhan and to become more infectious. Mutations of viruses are always in the direction of greater effectivity, not necessarily lethality, but infectivity, because natural selection favors the most infectious mutation because it reproduces the fastest. And what's very interesting, if you look at the US experience, the original outbreaks outside Seattle, in LA, and in Chicago were all from travelers who had come directly from Wuhan. I remember the index case in Chicago well. It was a couple that just returning from visiting their family in Wuhan. But there was a major mutation that took place in Europe and was responsible for the pandemic in Europe. And it was naturally carried by airline passengers to New York. There was a second mutation, G14, 614, which occurred later, which was the predominant infection in South America and is now probably the predominant strain worldwide. However, because of the fact that there is a particular choice of spike protein that people are designing the vaccines around, the vaccines under development do not seem to be affected by the current mutations. And a couple of days ago, Francis Collins, the director of the NIH, said it's a fairly well-behaved virus that we ought to be able to tackle effectively with a vaccine strategy. So far, the vaccine has not mutated too far away, the virus has not mutated too far away from the vaccine strategies that are being employed. But this is going to cause a huge disruption in the vaccine market. Our ideal goal is to have 7 billion additional people vaccinated for COVID-19 in 2021. Most current cellular-based vaccinations, particularly the ones using inactivated virus, require 2 injections in order to get full potency. Now, all the manufacturing process to make 14 billion doses has to be largely incremental because we can't stop making the other vaccines, particularly the childhood vaccines like measles and polio. So where are we going to get the capacity to make 14 billion additional vials? And another question is, where are we going to get 14 billion vials and stoppers? So we've got issues not only of manufacturing the vaccine, but manufacturing the stuff necessary to make the vaccine and to put it into a usable form. Now, some new vaccines, the messenger RNA vaccines, are expected to be a lot easier and less expensive to manufacture. There was an article in the Washington Post yesterday that made the comparison. It said 1 liter of messenger RNA can be used to make 2 million doses. In contrast to a traditional inactivated virus vaccine, which requires 30,000 liters of inactivated virus to make 2 million doses. So it may one of the reasons why everyone is focused on these messenger RNA vaccines is because they may be a lot easier and less expensive to manufacture, and so they will scale up faster. Speaking of scaling up, we need capacity to make 14 billion doses. Who are the companies that can do it? Well, let's look at the traditional players who are involved in this whole COVID-19 vaccine effort. The US, Merck, Pfizer are the 2 biggest by far. Janssen, division of J&J, is also very large. And we have a lot of smaller companies like Emergent Biosolutions, Novavax, Inovio, and Moderna, who are partnering with these large companies. Other large companies are GlaxoSmithKline, the UK-based firm, AstraZeneca, the British-Swedish firm, Sanofi in France is one of the world's largest vaccine makers. They make probably at least half of all the flu vaccines. The world's largest vaccine maker, however, is the Serum Institute of India, and they primarily make childhood disease vaccines for the Southeast Asia, Africa, Indonesia type markets. Sinovac, CanSino, and Sinopharm Fosun are 3 huge makers in China. And then in Japan and Korea, you also have significant makers. And there is one company, CSL Limited in Australia, which undoubtedly will be involved in manufacturing. Now let's look at that footnote. I've identified 4 companies, Novavax, Janssen division of J&J, Moderna, and AstraZeneca, who've all received funding from the US Biological Advanced Research and Development Agency, or the new Warp Speed grant. And it's about $3 billion total, but they are to enable them to help take the risk to do the scale up. So let's take a look just at one example of a collaboration. AstraZeneca collaboration with Oxford University. Basically, AstraZeneca is overlapping the startup of their production of the Oxford vaccine with the phase 3 clinical trials. So clearly the manufacturing startup overlap assumes that the Oxford vaccine will have a successful phase 3 clinical trial. AstraZeneca is taking the risk with the goal of saving 6 to 12 months in production startup. And they have received substantial commitments and now are setting the goal of producing 2 billion doses next year. The US committed 1.2 billion in funding to purchase 300 million doses. The European Vaccine Alliance has also committed about 400 million doses. And the Gates Foundation, which is a huge supporter both of WHO, about a third of the WHO budget, and the UN Child Vaccination Program, committed for another 300 million doses to be distributed under WHO charity programs. So, in addition, AstraZeneca has licensed the Serum Institute of India to make another billion doses to supply low and middle income countries, primarily in Asia and Africa, in Oceania. So the Oxford vaccine right now is the furthest along. So let's take a look at it in terms of the timeline for the vaccine trials and startup. So first of all, the phase 3 clinical trials are supposed to be complete by the end of year 2020. And that includes the probably close to 10,000 patients in the UK and assuming at least 9,000 patients each in South Africa and in Brazil, both of which are experiencing very large epidemics. AstraZeneca has risked financial risk, which has been backed by obviously grant commitments and manufacturing commitments, allows them to take a risk on startup prior to approval. Now you can make the vaccine, but you can't use it or sell it until you get regulatory approvals. In this case, regulatory approvals from the FDA and regulatory approvals from the European agencies and other major countries as well. The FDA has responded by creating this fast track program to speed up the actual approval, a final approval of the COVID-19. Now, to do that, you got to submit the results to the FDA of your phase 3 trials. That's typically about 60 banker's boxes, but I think it'll all be done electronically today. You're typically going to need 30 to 60 days preparation, let's say 30 days preparation, to get all the submission in order to the FDA. A maximum of 60 days has been guaranteed by the FDA under the Fast Track program. So if you add up the numbers, end of year for phase 3, 30 days to do the preparation for the submission. And remember, the FDA has been monitoring the trials the whole way, so there's no surprises here for them. And 60 days max for approval decision. I frankly think the earliest you would see the Oxford vaccine available for widespread distribution is probably early in 2021 in second quarter, maybe a little earlier in first quarter, but forget everything that you've read that says it's going to be available by the end of the year, because the phase 3 trials won't even be finished by then. So is that going to be, is the game over once that they get the vaccine? No. I want to point out that the first product to market is not necessarily the winning product in the marketplace, because real long-term success will depend on a number of technical and medical and market factors. Most important are the degree and length of protection which is provided and by the vaccine. Okay, is it 80% effective? Is it 90% effective? Ideally 100%. And will that infectivity last for the rest of your life, like chickenpox? Or is it something that you're going to have booster shots every year? And boosters, and you're going to have new vaccines developed every year to cope with mutations. Insensitivity to the virus mutation is a huge technical characteristic of a successful vaccine. The other thing is the messenger RNA vaccines may be a single dose. Okay, there's certain recombinant DNA vaccines that are used to create B cells, which are long-lasting cells which will produce antibodies. And they may be a single dose vaccine. But right now, the vaccines which are being developed to an activated virus are at least 2 doses, and we don't know whether boosters will be necessary. And as Mohammed and I can tell you, okay, vaccines which don't require refrigeration are the holy grail of vaccines, because it is so hard to find appropriate refrigeration support in Africa and Southeast Asia, that the cold chain support is critical to the current childhood vaccines. So what other candidates do we have that may be second or third or 10th? Okay, there are 7 candidates that the Chinese have in clinical trials or about to go into it, of which at least 4 of them are very strong candidates. We talked about the CanSino vaccine, which has already been distributed by the People's Liberation Army. And there are very large production capacities able to support vaccines at Fosun, Sinopharm, and Sinovac, which are already large vaccine manufacturers. The messenger RNAs may be the most technically advanced. They may be easier from a manufacturing point, assuming you have to develop all new processes, but they're just starting phase 3, and at this stage we have no information, as to what the outcome will be. So the last point is not my opinion, it's Dr. Anthony Fauci's opinion. He gave an interview last month with the Daily Telegraph of London. And he looked at the vaccine landscape and he said, we have potential vaccines making significant progress. We have maybe 4 or 5. And he went on to disclaim that you can never guarantee success with a vaccine, and it's foolish to do so because there's so many things that can go wrong. His bottom line is everything we have seen from the early results, it's conceivable that we get 2 or 3 vaccines that are successful. So that is basically a summary of the program and where it stands. And at this stage, maybe we can go to the next slide and take a few questions. I may not be able to answer them, but I'll certainly tell you what I can.
All right, thank you, Clive. Appreciate your insights and information you brought to us today. And we do have some questions, so hopefully we can answer some of those to the community here on the call right now. The first one we have, and this relates to AIDC, I think that many would be interested in what they can do to help with the COVID problems. What do healthcare community members need that AIDC can provide?
I think that's a very good question. All of these are going to need labeling issues. There's going to be a lot of, they're very small vials in typical cases, going to be a lot of very small 2D labels that will be used. You need to be aware of what the labeling standards are, particularly in Europe as well as the US. Unfortunately, right now, we're still in the process of sorting out all the drug labeling stuff, but you need to stay aware of it. You need to be prepared to support it with equipment and with for both labeling and field reading of these, because remember, most of the places this is going to be used, okay, don't have barcode readers. And some of the technologies which are being developed by Temptime, now part of Zebra, okay, are also going to use sensor barcodes. And that is something that requires some special reading technology. And you can contact Dr. Mohammed Abdu or me, and we will be happy to put you in touch with it. But development of technology for labeling and for particularly reading sensor labels in the field may be a very interesting opportunity as well as a necessary technology. Thank you.
Yep. Now, Clive, you touched on this a little bit during your presentation, but when a vaccine is ready, or if vaccines are ready, how long do you think it will take for it to be widely available? And do you believe these vaccine makers will be able to handle the demand?
Handling the demand, I believe, will happen. One thing that I've gleaned from all the reading is that the vaccine manufacturers are investing money in the manufacturing of the vaccine that they are supporting. And they are already scaling up projects. They're building new equipment, new plants, new production lines. So I think the vaccine manufacturers at least will be able to produce maybe 2 or 3 billion doses next year, okay, because they've invested risk capital and they are each of them focused on a particular candidate. They're going to be people that lose a lot of money too, let's be honest, but it's risk reward. In terms of distribution, this is what God gave us airplanes for. And I think you're going to see global distribution. You're going to see global manufacturing very quickly. Okay, many of these people have individual vaccine candidates have multiple organizations supporting them. They have partners. They've contracted with companies like Serum Institute of India okay, which is a very big manufacturer, to produce it for countries that they may not be able to directly distribute in. So I think the distribution phase will be fairly quick. The biggest thing is going to be approval and scale up of the manufacturing.
Okay, so one of the other questions we received, and it kind of goes along with what you just said, is who will be producing the vaccines if one becomes available? Are all drug companies allowed to, or do they need to get a license or approval to make them?
And they need to be licensed because the the people, let's take AstraZeneca Oxford. Okay, the AstraZeneca partnership with Oxford basically gives AstraZeneca the rights, but it also to manufacture. But AstraZeneca has been smart enough to realize that even though they're huge and multinational, they don't distribute and they don't manufacture in certain key areas like Southeast Asia and Africa, whereas Serum Institute of India does. And so they license Serum Institute of India to make it for those marketplaces. So I think that's pretty much what's going to happen. You're not going to have everybody making everything. There are going to be licensing arrangements. Vaccine manufacturers do have to recover their investment. And this is not something, I don't think it's going to be something that is thrown into the public domain because nobody would ever make the investment because they can't recover it.
Okay. Another question we received is, are you aware of the latest on whether those that survive the virus can get infected again?
No, and I think that is a huge question for the vaccines, and it's something that may not even be answered in the clinical trials. You may be able to prevent some people from getting it, but we're finding right now that even some of the therapies, antibody therapies and stuff that they're giving them are no guarantee that people won't get infected again. And the real question is, how long is it going to be effective? Is the vaccine going to be effective for a year? Is it going to be effective for a lifetime? That is an unanswered question that only can be done with years of experience.
Okay, thank you. And I think this goes along with it as well, what you just said. What are some of your thoughts on the antivirals and monoclonal antibodies out now, and how do they factor into the development of a vaccine?
The second part of it, I really can't answer that question. I really do not. I have not followed that, and I do think that some of the drugs. Let's talk about one drug in particular. Okay, remdesivir. Okay, from Gilead Sciences, it's shown definite effect to shorten the length of the infectious period, but it seems to be a lot like the first drugs we used to treat AIDS. A single drug is probably not going to be the solution with coronavirus, and it certainly wasn't the solution with AIDS. The HIV drugs, it takes a combination of 2 or 3 drugs because of the fact that the virus has many modes of action in the body. And so I think there are very promising drugs. Regeneron has just started a new approval trial. But we don't know what the results of these are going to be. And while these potential drugs are very interesting, no one's had enough experience to say, is the drug itself going to be effective, or is the drug in a combination with other drugs going to be effective? So it's a great question, and I think it's probably going to be a year before we have the answer.
Okay, thanks. Another question we received, which is more US based, is what is the likelihood that vaccines are fully covered by insurances in the US? And also, what's the likelihood that a sufficient population in the US would be able to take it, both financially and willingly?
That is a big debate, and Dr. Fauci just weighed in on the last part. First of all, since the US government is funding to the tune of about $3 billion the development and startup of some of these vaccines, okay, not only the US effort, but the Oxford effort, one would hope that Medicare and Obamacare would cover the cost of the vaccine in the patient. Certainly, they do. Medicare covers influenza vaccine on that. So one would expect that to happen, and I'd be very shocked if it didn't. In terms of what the public reaction is going to be, this is where Dr. Fauci observed yesterday. We got a lot of people in the US who basically don't trust the government. They've been told that COVID-19 is a hoax, and therefore, why would they want to get a vaccine for it? We have a lot of complete anti-vaxxers, okay, when we're losing our herd immunity in the US to measles, because measles requires about 95% vaccination to have a herd immunity, because it's about 18 times more infectious than COVID-19. So it's going to be a very interesting issue as to whether people will sign up to be vaccinated. I and my wife are in a very high risk group. It will take me about 0.2 nanoseconds to make the decision to get vaccinated because of that, but there are a lot of people who are going to be very resistant to vaccination just for the same reasons they've been resistant to compliance with the mandates on social distancing and masks, etc. Don't know how that's going to play out, and it's very worrisome.
Okay, and we'll just leave with one last question here, and that is there concern about potential side effects from these vaccines, and will the very vulnerable be able to deal with those?
That's an extremely good question. Supposedly, the phase 1 and 2A clinical trials should show whether there's significant adverse effects, but as to whether there are long-term effects, only time is going to tell. Thank you all. I see many friends out there, and I greatly appreciate you bearing with me.
Thank you, Clive, for your time today, and audience, thank you for your active participation. I hope everyone stays safe.